Hello everyone, and topic for today's discussion is plant tissue culture. Introduction. Plant tissue culture is an in vitro culture or growth of cells, tissues or organ of plant in a sterile condition and well formulated media to produce an entire plant. The controlled condition provide the culture an environment conducive for their growth and multiplication. These conditions include proper supply of nutrients, pH medium, adequate temperature, and proper gaseous and liquid environment. Basic terms used in tissue culture. Explant. It is an excised piece of differentiated tissue or organ is regarded as an explant. The explant may be taken from any part of the plant body, example leaf, stem and root, etc. Callus. It is an unorganized and undifferentiated mass of plant cell is referred to as callus. Generally, when plant cells are cultured in a suitable medium, they divide to form callus. De-differentiation. The phenomenon of mature cells reverting to meristematic state to produce callus is de-differentiation. Re-differentiation. The ability of the callus cells to differentiate into a plant or organ, a whole plant is regarded as re-differentiation. Totipotency. The ability of an individual cell to develop into a whole plant is referred to as totipotency. Coming to the steps involved in plant tissue culture. First is preparation of suitable nutrient. Generally, MS medium, White's medium, B5 medium or few culture media that can be obtained from the market for the lab preparation of nutrient medium needed for tissue culture. The culture medium consists of inorganic nutrient. We have two types of inorganic nutrients are used in the culture medium. It consists of 6 macronutrients like nitrogen, phosphorus, sulfur, calcium, potassium and magnesium, and 6 micronutrients like iron, molybdenum, copper, zinc, boron and manganese are also added in the medium. Organic nutrients. Carbon and nitrogen containing compounds are used. Sucrose, fructose and other carbohydrates are the carbon source. Vitamins and amino acid are the nitrogen source. Auxin, cytokinin and gibberellin are the most commonly used growth hormone in culture medium prepared for tissue culture. Agar. It solidifies the medium and acts as a substratum. It does not have any nutritive importance. It is obtained from seaweed and it is used to provide solid surface for growth. All these important nutrients are incorporated in the culture medium and transferred into suitable container and then sterilized. Select of explant. Explant or any excised part of the plant to be used in tissue culture like axillary bud, leaf and stem segment, root tip, shoot tip, anther, ovary and endosperm. Always young and healthy part of plants are selected as explant. Third step is sterilization of the explant. Explants are sterilized by using different types of disinfectant like sodium hypochlorite 2%, silver nitrate 1%, calcium hypochlorite 9 to 10%, bromine water 1 to 2%, mercury chloride 0.1 to 1%, hydrogen peroxide, etc., and different types of antibiotics. Fourth step is inoculation of explant. The sterile explant is inoculated on the surface of the solidified nutrient medium under aseptic condition. Laminar airflow can be used for this inoculation process. And next step is incubation. Incubation is done at 25 plus or minus 2 degrees Celsius with a relative humidity of 50 to 60% with 16 hours of photoperiod. After defined period of incubation, an unorganized and undifferentiated mass of cell called callus is obtained from each explant. In some cases, root and shoot directly develop from the explant after incubation. Sixth step is transfer of callus and regeneration. The callus obtained from incubation of explant is transferred into another medium and re-incubated. Plantlet regenerates from the callus with the induction of root and shoot directly from the callus. The next step is hardening and acclimatization. Hardening is a gradual exposure of the plantlet for acclimatization or adjustment to the environmental condition. And last step is the transfer of plantlet to the feed. After the hardening process, plantlets are transferred to either the greenhouse or the field condition outside. And these plantlet will grow into independently and healthy plants. Coming to the factors affecting tissue cultures, we have three factors: growth media, environmental factor, and explant source. Next is types of tissue culture. We have micropropagation. It is the rapid vegetative propagation of plant under in vitro condition. Next is germplasm conservation. Conservation of germplasm involves the preservation of the genetic diversity of a particular plant or genetic stock. And third type is somoclonal variation. The genetic variation found in the in vitro cultured cells are collectively referred to as somoclonal variation. Next step is haploid and dihaploid production. In this plant which possesses only a single set of chromosome in their sporophyte are known as haploid plants. And next is in vitro hybridization, protoplast fusion. In this technique which allow the manipulation of cellular genome by protoplast fusion. And next step is embryo rescue. It is an in vitro technique used to development of an immature embryo into a viable plant. And last type is synthetic seed production. In this, seeds produced by encapsulation of somatic embryo. And coming to the advantages of plant tissue culture, it's a fast commercial propagation of new cultivars. It's taking an explant does not usually destroy the mother plant. So rare and endangered plant can be cloned safely. Once established, a plant tissue culture line can give a continuous supply of young plant throughout the year. The production of clones of plant that produce particularly good flavors, fruits, or have other desirable trait. The regeneration of whole plant from plant cells that have been genetically modified. And these are all some references. Thank you.